Hi everyone, my name is Ieritano and I am a PhD student in the Hopkins Group in the Department of Chemistry. Today I'll be discussing the works of myself and my colleagues that combines machine learning with differential mobility spectrometry to improve drug design. So in the primary stages of the traditional drug design process, libraries consisting of thousands of compounds are screened for their affinity towards a particular biological target. Once a hit that binds to a target is identified, it must be evaluated for numerous properties that determine how well the drug will perform in the human body, such as its ability to cross a cell membrane. Even with high-throughput analyses, the early phase testing portion of a potential drug candidate can take up to 6 years. This is an extremely long time considering the rising state of antibiotic resistance and the global pandemic that's currently underway. It's clear then that we need a way to speed up this preclinical testing portion in order to bring therapeutics to market faster. Since we know that the properties that determine a drug's performance are ultimately related to the interaction of the drug with its solvation environment, we can potentially correlate drug function with its innate solvation propensity. To do this, we use a technique called differential mobility spectrometry. While this technique is traditionally used as a gas phase separation technique, the chemistry that goes on within the DMS cell is exactly what we need in order to mimic a dynamic solvation environment. A rapidly oscillating electric field drives cycles of condensation and evaporation of water around a drug candidate. The motion of a drug molecule as it travels through the DMS cell correlates strongly with the drug-solvent interaction, which in turn correlates with common properties that are screened during drug development. DMS measurements, which are supported by computational models, serve as inputs for custom machine learning tools that can further strengthen our ability to predict properties sought by pharmaceutical companies. As these measurements require small amounts of material and take minutes to complete, we can rapidly generate physicochemical property data that defines a drug's viability for human consumption. Our preliminary assessment of this method is the subject of a patent and a publication in Nature Communications, where our AI-driven approach yields predictions on the y-axis that correlates exceptionally well with traditional measurements on the x-axis in a fraction of the time. Beyond drug property prediction, we can also use AI tools in DMS data to repurpose drugs that already exist. AI-driven computational screening of roughly 500 approved drugs belonging to 5 different pharmacological classes assigns these drugs according to the prescribed function with about 94% accuracy. However, it's the off-axis points in this correlation matrix that are particularly useful, as they may indicate new functionality or potential side effects of existing drugs. And with that, I will extend my thanks to my supervisor, Scott Hopkins, his spin-off company, Watermine Innovation, my lab mates, and our various funding sources.